share my research endeavors. So here I will be focusing on some of my ongoing research work on the treatment of cancer. So as you all know that the worldwide tackling cancer still remains a daunting task for clinicians and researchers. So among different types of cancer, lung cancer is predominantly associated with the highest mortality, followed by the liver cancer. So it is generally estimated that by the year 2025, globally, there will be a surge in the number of cancer cases, more than 20 million annually. So this alarming statistics has compelled the researchers across the globe to expedite the research for newer and potent molecules to overcome the different type of the cancer. However, as you all know that like the most of the existing anti-cancer agents, especially the chemotherapeutic agents, like they fail to discriminate between the cancer versus healthy cells, and this leads to a lot of toxicity issues. So keeping this into fact into consideration, people are opting for like some alternate therapy. So in that regard, like it has been found that the small interfering RNA, which are also known as the short interfering RNA or gene silencing RNA, they are exploited by the nature for post-translational gene regulation strategy. So it has also been found like if administered from outside, these siRNA can interfere with the expression of specific genes which are responsible for propagation of the cancer. So considering this fact, like siRNA and also the CRISPR-Cas9, they are like recently being tried for treatment of cancer. So in the RNAi involves the multi-step process where the exogenous RNA is administered, which is in the form of the double stranded form. So consisting of the passenger strand and the guide strand, and this passenger strand is degraded and the guide strand is incorporated into RNA-induced silencing complex. So the most well-studied outcome is the post-transcriptional gene silencing, which occurs when the guide strand base pairs with a complementary sequence in the messenger RNA molecule and induces the cleavage by the argonaut. However, even though just like the other chemotherapeutic agents, the selective silencing of the gene expression using siRNA has been investigated as a potential therapeutic approach to treat cancer. However, because of the large molecular weight and also the polyanionic nature, naked siRNA does not freely cross the cell membrane. And if, however, it somehow crosses the membrane, so this is having some specific fate where like enzyme present in the systemic circulation, they generally degrade the siRNA, and this will be leading to the like ineffectiveness of the siRNA. Just to overcome like these issues, like people across the globe, like they are using some specific type of the drug delivery vehicles, where this siRNA is being encapsulated into various nano-sized vectors, and it has been found the chemical mediated delivery or the electroporation or the like incorporation of the siRNA in the viral vectors and also nanoparticles, it will be just overcoming like some of the issues which are associated with the non-effectiveness of the siRNA in the clinical setting. So here we will be focusing only on the lipid-based nanoparticles and also on the inorganic nanoparticles. So where we are using the conventional liposomes and the bacteriosomes. So I'll be focusing on the work where we used stereosome-based cytosolic delivery of the polo-like kinase specific RNA, and we assessed the potential of this delivery system in treatment of liver cancer in biopsy mice. And in the subsequent studies, like we also encapsulated this siRNA in dendrosomes, or maybe like we made a dendrimer PLGA based multifunctional immuno nano composite where we encapsulated doxorubicin in the PLGA based microspheres while siRNA was entrapped in the dendrimer, and on the surface of this whole composite we put some cancer specific antibodies. And we also exploited like specific use of the like some of the biological systems for fabrication of the nanoparticles, and we use like these nanoparticles for treatment of the fibrosarcoma in the model animals. So I'll be studying on the first component where like we use stereosomes. So as the name is indicating, the stereosome we are consisting of the basically lipids which were isolated from the plasma membrane of the prokaryotes, that is the E. coli and the Bacillus subtilis. We isolated this plasma membrane lipid by Blyodire process, and then we use a dried reconstituted vesicles for entrapment of the siRNA inside the liposomes, where the plain liposomes were first prepared by the sonication method, and then they were just mixed with the solution of the siRNA. Here you can see that a specific amount of the siRNA was trapped inside, and then the as formed nanoparticles, they were characterized for their surface properties. So here we are just sharing with you the representative transmission electron microscope image of the stereosome based nanoparticles. You can see here the like TEM analysis and also the corresponding particle analyzer data of the stereosome which we are harboring the siRNA, first ascertained by the nanofox system. And then we also determine the size of the as formed nanoparticles using the atomic force microscopy. So all these three techniques were used for determining the size of the particles, and we found like the size is ranging between the 100 and 150 nanometer. Then we determined the release of the siRNA from the parent stereosome based nanoparticles, and we found that the maximum release is happening in case when EC siRNA are being suspended in the solution which is having pH 5. And you can see the extended release of the encapsulated siRNA for up to the 50 hours. Then we administered like we made like different preparation where we encapsulated the siRNA in the stereosome, and then we use like this stereosome bearing siRNA bearing stereosome for treatment of the cancer which was induced in the model animals by their exposure with the diethyl nitrosamine chemical exposure. And here you can see the histopathological studies showing the effect of PLK1 bearing specific siRNA bearing stereosome on the liver cell, which is already having the liver cancer. So you can see like the group of animals, that is the cartoon E, it is showing the effect of the stereosome based siRNA nanoparticles on the liver cells with intact lobular architecture, hepatic laminae, contour staining of the hepatocyte is markedly visible. So among like different formulation, stereosome based siRNA treatment gives the best recovery in diethyl nitrosamine induced necrosis compared with the free siRNA group. So next we determine the effect of this as formed siRNA bearing stereosome on the expression of various protein which are responsible for regulation of the cell cycle of the cancer cells. So here we look for the expression of the wild type P53, and as you can see in the cartoon that that group of the animals which was treated with the siRNA bearing stereosome, then it was showing the concentration of the P53 wild type molecule, it is being just revoked as compared to the group which was treated with the free siRNA. In contrast, the concentration of the mutant form of the P53, it is less in the group which was treated with the stereosome bearing siRNA. Then the effect of this treatment on the expression of the Bax protein was also seen, and you can see here the expression of the Bax protein is revoked, the group of animals which was treated with the EC siRNA nanoparticles. Then we also determined the effect of the this EC-siRNA nanoparticle treatment on the expression of the polo-like kinase. As you know that the polo-like kinase is responsible for the proliferation of the cancer cells, especially the liver cancer cells. So we determined the effect of this siRNA on the expression of the PLK1 because this siRNA was specific for the PLK1 enzyme. You can see here the expression of the PLK1, it is just getting downregulated in the group of the animal, which was treated with the stereosome, which were containing the specific siRNA as compared to the free form of the siRNA. Then we also determined the effect of the this siRNA bearing stereosome treatment in induction of the apoptosis in the cancerous hepatocytes, and where we look for the expression of the important pro-apoptotic enzyme caspase 9. As you can see here, like in cartoon, the animals, the liver cells of the animals which were treated with the siRNA bearing stereosome, they are showing predominant expression of the caspase 9. Then we also determined the effect of the like this siRNA treatment on the expression of the liver cancer treated hepatocyte isolated from the, so where we perform the tunnel assay, and you can see that in the tunnel assay the ApoBRDU tunnel molecule, like it is more abundant like in the group of animals like which was treated with the PLK1 specific siRNA bearing stereosome. Then we also determined the effect of this siRNA bearing stereosome on expression of the annexin 5, which is a like molecule which is expressed on the surface of the apoptotic cells. And as you can see here, the expression of the annexin 5 was more predominant the group of animals that was treated with the stereosome based siRNA nanoparticles. So on the basis of this study, we can conclude that the anti-cancer efficacy of the stereosome can be increased by its encapsulation in the stereosome. This stereosome we are prepared by the lipids which were isolated from the plasma membrane of the E. coli. So as you know that the plasma membrane of the E. coli, it is full of the anionic lipids, and these anionic lipids are basically your phosphatidylethaноlamine, cardiolipin, and phosphatidylglycerol. So in contrast to the mammalian cells or the like plasma membrane of the other type of the living cells, the predominance of the phospholipids, like which is your phosphatidylethaноlamine, phosphatidylglycerol, and cardiolipin, this is responsible for the rapid membrane fusion, which is can be considered as a factor responsible for to cope up with the high multiplication rate of these prokaryotic cells, which have got the generation time of the 20 minutes. So in contrast to the like other type of the living cells, like for example your mammalian cells, like they are taking almost 24 to 30 hours like for their duplication, and again like your Saccharomyces cerevisiae cells, like which are having a combination of both phosphatidylethanolamine and also the phosphatidylglycerol and the phospholipids such as sphingomyelin and the PC, like they are having generation time of the order of 2 hours. In contrast, the prokaryotic cells, like for example your Bacillus subtilis and your E. coli, like they are just multiplying, okay, and duplicating in very short time period. And you can see like the presence of the like specific phospholipids, like for example your cardiolipin in the intruding septum and also on the corner in the dividing cells. So for considering this very fact, like we thought of like isolating lipids from these what you call microbes, and then we prepared liposome out of them. So in the next study, like we encapsulated this PLK1 specific siRNA, the dendosomes, and we determined its efficacy against the skin tumor. Okay. So in the next, like we combined like this. Quickly, I'll be going through that. The next, like we made a nano composite, which was consisting of the like PLJ microspheres, where the PLJ microspheres like they were loaded with the cisplatin anti-cancer drug. In contrast, like your G4 palmam dimers, like they were loaded with the PLK1 specific siRNA, and then we made like a composite of these two, that is your PLJ based macrospheres and the dendrimers, and then on the surface of these dendrimer PLJ nanocomposite, we immobilize the tumor surface specific antibodies, and we demonstrated that the antibody conjugated PLJ dendrimer nanocomposite complex to siRNA can enhance the efficacy of both free form of the cisplatin as well as free form of the SIR. So in the next study, like we encapsulated like this siRNA in the specific type of the liposomes, which we named as the subtilosome, and here we isolated lipids from the plasma membrane of the microbe Bacillus subtilis. So the bacteria was grown, and from at the log phase particle grown culture, we isolated lipids, and then same way like we prepared liposome, and we loaded this empty liposome with the free siRNA, and we prepared a like siRNA pairing subtilosome, and then we determined the specific characterization of the like this as formed subtilosome, which were harboring the PLK1 specific siRNA. So here you can see like the subtilosome were characterized on the basis of the TEM analysis again, also on the basis of the their specific distribution by the was determined with the help of the nanophosphatizer, and we also determined the release of the encapsulated siRNA, and then finally we determined the effect of these specific COX-2 siRNA specific ribosome against skin cancer. So here you can see the effect of the cancer on the liver cells, okay, and how like when it is treated with the like your specific subtilosome bearing subtilosome which we are bearing the siRNA, then it will be just able to subside the cell cancer in the liver cells. Then it is the like survival graph of the different COX-2 siRNA treated animals. See here. So finally, like we developed a specific formulation of the 5-FU, which is a very potential anti-cancer agent, and because of the again some intrinsic toxic properties like the wide usage of the 5-fluorouracil is restricted. So here we developed 5-fluorouracil nanoparticles, okay, and then these nanoparticles like they were not having any excipient. So these nanoparticles were just fabricated with the help of the orange juice. So we established that certain components present in the orange juice, they can mediate the development of the 5-FU nanoparticles, and these nanoparticles like they are free of any excipient. And as the excipients, like these are the limitations, like in terms of the various particle aspects, like because they are different type of the excipients like the phospholipids and the like PLGA and some other type of the like excipients like which are used for the development of the. So they are making the like the as formed nanoparticles cost effective, and they are also having full of like different type of the toxic intrinsic toxicities, and then just to overcome this problem, like we used like 5-fluorouracil as a parent drug, and it was just treated with the orange juice, and we found that like it is leading to the formation of the small sized nanoparticles which are having a size dimension less than 100 nanometer, and this is the AFM and the electron TEM analysis. And then you can see here like we further characterize it for the purity and stability of the as sensitized 5-FU nanoparticles, implying the FTIR, DSC, and the XRD analysis. Then we determine the effect of these as formed nanoparticles on the expression of the various factors which are responsible for the progression of the cancer. Here you can see like the expression of the p53 wild type. It is just upregulated upon treatment with the 5-FU nanoparticle. In contrast, the expression of the 553 mutant form molecule, it is downregulated, and effect of the expression of the BSCL2, like it is also downregulated in the animals which were treated with the 5-FU nanoparticles. However, the expression of the BAX was upregulated. Then we again determine the effect of this treatment on the caspase-9 expression. You can see here the caspase-9 expression is more in the cells like which were the cells which were isolated from the animals which were treated with the 5-FU nanoparticles. Then we also determine the purposes on the basis of the tunnel assay, where we just look for the expression of ApoBRDU analysis. Again, like you can see the animals which were treated with the 5-FU nanoparticles, they are showing more fragmentation of the DNA. Then we also determined the effect of the these 5-FU nanoparticles on the expression of the Nextin-5, which was expressed on the apoptotic cells, and you can see here the expression was around 47% in the animals which were treated with the 5-FU nanoparticle, in contrast to the animals which were treated with the free form of the 5-FU nanoparticle, where the Nextin-5 expression was only 33%. Then we saw the effect of these nanoparticles on the skin fibrosarcoma. Here you can see there's a great deal of regression of this fibrosarcoma in the animals that were treated with the 5-FU nanoparticles. The overall survival rate was also increased, and the tumor volume was also less in the animals which were treated with the 5-FU nanoparticle as compared to the free 5-fluorouracil. And you can see here the histopathological analysis. Again, like it is showing that the animals which were treated with the 5-FU nanoparticles, like they are attaining the more features just like similar to that of the healthy animals. So on the basis of this study, like we can conclude that the cellular drug delivery vehicle, including the natural as well as the synthetic system, have potential to specifically deliver the encapsulated siRNA to the specific cancer target site, and then they are also retained because of the higher efficacy, as it is indicating that the more EPR effect for the nanoparticles which are conjugated with the nanoparticles or which were developed as a nanoparticle. So the synthetic carriers basically deliver the synthesized siRNA as well as the other chemotherapeutic agents, and then this can be specifically targeted to the cancer cells only. So by opting for nanoparticle mediated siRNA and chemotherapeutic agent delivery, we are optimistic about dealing with the dreadful diseases like cancer. So with this, thank you to you all for the patient hearing. And also thankful to the organizer for giving me the opportunity to share my research endeavors. If you have any question, please ask.